So last time we have discussed this classification. We have discussed about implicit memory. Now our focus mainly will be on explicit memory now. Okay, explicit memory all we have already studied. Okay, now what are the various structures? How does exactly explicit memory occur? Okay, molecular mechanism to molecular mechanism we will come back afterwards. Okay, so our now focus is how does explicit memory is occurring. Okay, so see, before going I have already told you that in implicit memory, mainly you can say implicit memory is for the skill. Okay, in implicit memory these structures which were involved were other structure other than hippocampus and what, temporal lobe, you can say medial temporal lobe. These were the structures which were playing main role. Okay, there in implicit memory. Okay, now in explicit memory this hippocampus and medial temporal lobe, these are playing main structure. We have already discussed in thalamus. We have already discussed in thalamus one circuit known as Papez circuit. What was that circuit? Papez circuit. Okay, now I will just draw in flowchart form. Okay, like see, okay, what I will do, I will just tell you exactly how does this memory is stored. Okay, so this is your lateral view of cerebral cortex. Okay, now whenever you are getting new information, okay, you are getting a new information, you can getting means your visual things, auditory, all things will from everywhere you are getting new information. So initially this area is there. Okay, you can call it as prefrontal cortex. Initially it is there in prefrontal cortex. Okay, then what is there from here? See, this was number 1. Okay, then there is one structure here. This is a structure of temporal lobe, or specific you can call it as hippocampus. Okay, then it will go to hippocampus. Okay, then from hippocampus there was one structure known as fornix. Through fornix, where it was going? It was going to thalamus, anterior nucleus of thalamus. So whenever new information comes, first it will go in the prefrontal cortex, second hippocampus. Okay, now why hippocampus? Hippocampus has a vast connection to limbic system. Okay, it has vast, so it is related to emotions. Okay, so whenever you want to learn new things and it involves emotion, it involves motivation, you can remember it easily. So whenever you are studying anything, you have to study with the interest, you have to study with the motivation. If you are not studying with the motivation and interest, you will find it difficult to learn. So that is how this is playing the role. Okay, so initial information has come, initially it has gone here, from here it is coming to hippocampus. Okay, so from hippocampus it has gone to thalamus. Okay, from thalamus where it will go then? From thalamus it will go to, you can say, cingulate gyrus. Here I am not drawing it, it is it is like this, cingulate gyrus. Okay, then from cingulate gyrus mainly other structure is there, entorhinal cortex, like here it is coming, and again it was going to hippocampus. Okay, so that was Papez circuit. Okay, now here not only this, from here what is the from cingulate gyrus, from here information is going to where, you can say basal forebrain. Basal forebrain is not, means here only basal forebrain it is going, cingulate gyrus, entorhinal cortex. Okay, so in the basal forebrain, basal forebrain means part which is situated at the base of the brain. So there is one nucleus known as nucleus basalis of Meynert. So what is there? It emerged from here, cholinergic neurons are there. So cholinergic neuron will send signal to whole the neocortex, or you can say various association cortex. So what I meant to say that, see, initially what was there, information has come to prefrontal cortex, then hippocampus. Okay, so if you are reading it again, if you are studying in detail, ultimately through all the structure from basal forebrain, information will be stored to various large areas of neocortex, widespread area you can say. It will be stored in the widespread area of neocortex. Okay, so that is how it is there. Okay, so whenever any new information is coming, gateway is what? Gateway is mainly hippocampus. From hippocampus only it will be stored. Okay, so that is how is anterograde amnesia. In anterograde amnesia, you will not able to learn new things. Okay, like you will be able to learn, but after some time, see, this information will not go to neocortex. So that is how that you can say consolidation will not be there. Okay, so hippocampus and temporal lobe, so they are playing role in explicit memory through this mechanism. Okay, now see here, so explicit memory here, which are the main, so hippocampus and middle temporal lobe was playing the main role. Beside this, here amygdala is also there, which is playing important role through emotions. Through what? Emotions. Okay, so understood. So we have classified the memory, we have studied implicit memory and explicit memory. Implicit memory you have studied various structure, explicit memory you have studied various structure. Okay, so if there is problem with the hippocampus and temporal lobe, okay, what will be the problem? Problem will be what? Here patient will not be able to learn new things. Okay, so that is why it is known as anterograde amnesia. Okay, then there is some other term like you can call it as retrograde amnesia. Okay, what is retrograde amnesia? We will come back to it afterwards. Okay, so that is how we have classified the memory. Okay, we will just revise what we have studied depending on the time we have classified. Then again classification. Okay, structure, this is the time. Okay, now see, important, you can't separate right and left. Okay, because both are connected via corpus callosum. Both are connected via corpus callosum. So this is how this intracortical transfer takes place through the corpus callosum. Okay, so now we will be studying about the molecular mechanism which are playing the role in the molecular mechanism which are playing role in all these types of memory. Okay, so molecular mechanism we have already studied, habituation and sensitization. Okay, now we will be studying what are the mechanism for long-term potentiation and long-term depression. Okay, now this long-term potentiation, long-term depression. Okay, see, molecular mechanism, you can't just tell that it is separate for implicit and it is separate for explicit. Ultimately thing is that all these are occurring at the level of synapse. Okay, so in synapse all these mechanisms are occurring. Only thing what is occurring, okay, like in some case you can say in various means parts of brain will differ. Okay, in certain parts means like in means or whatever mechanism is there, it will be occurring in various other parts in implicit and various other parts in explicit. Okay, now if you take in explicit memory this long term potentiation, long term depression, beside that you might have heard post tetanic potentiation. All this, where you have studied all this, we have studied all this where in synaptic sorry no so we have studied all this in what, synaptic plasticity. Okay, so all these are nothing but the mechanisms of synaptic plasticity. So all these are the molecular mechanism which will play a role. Okay, so before studying this molecular mechanism I will be telling you in general how this you are memorizing. Okay, see, whenever you are learning anything for short term, okay, so initially some mechanism will occur at the level of synapse. Okay, if strong emotional components are there, then potentiation will be there, means long term potentiation or sensitization will be there. So from short term, then what will happen, ultimately it has to go to intermediate or long term. Okay, so whenever it has go to intermediate or long term, what will happen, other structural change. Okay, before structural change what will happen, more and more proteins will be there. Okay, more and more synaptic vesicles will be there, and ultimately in the end what will be happen, structural change you can see. Okay, and various nerve growth factors will play role. Even it is said that some cells, especially in you can say olfactory cell, if you take a part of brain in olfactory cortex and hippocampus, these are the two portions where new cells are formed. Okay, so ultimately because of all this mechanism, because of, see, over the short term molecular mechanism will occur, over the long term this molecular mechanism will ultimately reflect into a structural change, and structural change may be so huge that new cells may be formed. Okay, so these are the various molecular means these are the in general various changes which are occurring. Okay, now we will be studying about these two mechanisms, that is long term potentiation and long term depression in detail, how exactly they are occurring. Okay, so habituation we have studied sensitization. So this is a mechanism of this that was the mechanism of long term potentiation. Okay, so I just want to explain you this mechanism here, then we will go to that diagram. So in hippocampus, this is mainly occurring where, in hippocampus. In hippocampus there are again two fibers are there, one is Mossi fibers, another is Schaffer collaterals. Small difference is there, like if you say in Mossi fiber, okay, here it is, you can say NMDA independent. if you take Schaffer collateral, okay, here AMPA and NMDA, two receptors are there which are playing the role. Okay, mainly you can remember this Schaffer collateral. Okay, how exactly how this AMPA and NMTA, okay, the how it is not independent. This mainly depends on NMDA only. AMPA does not play any role here. Okay, so what is exactly this mechanism that we will study now. So sorry for that. Okay, so now I will be giving you this example. See, this is a presynaptic terminal and this is postsynaptic terminal. What it is? This presynaptic terminal, and where we are studying? We are studying in the Schaffer collateral. Okay. Now here see, two receptors are there. Here it is AMPA and here it is NMDA. N-methyl D-aspartate, AMPA, right now I am not recollecting it. Okay, so two receptors are there, one is AMPA, another is NMDA. Now whenever there is stimulus, see here, whether it is low frequency or whether it is high frequency, it also matters. If it is low frequency, then what will happen? LTD, long term depression will occur. If it is high frequency, what will happen? LTP, long term potentiation will occur. Okay, so what is occurring here now? Okay, so see, initially whenever stimulus is there, what is released? Glutamate is released. Glutamate will be released. Now glutamate, see, initially if you take NMDA, there is a block because of what? Magnesium. Okay, you can say magnesium block is there. Okay, so initially what will happen? This glutamate will go and bind here. Okay, now as a result of that, from here what will come? Sodium will come. What will come? Sodium will come. So first event will be what? Depolarization. Okay, so this depolarization, it will relieve this magnesium block. Okay, means magnesium will now go away. Okay, so see, AMPA first has stimulated. Okay, and as a result of that, it has occurred. Okay, now this is what Schaffer collateral. In mossy fiber, this AMPA event is not there. There mainly NMDA events are only occurring. Okay, now glutamate will do what here? Glutamate, see, magnesium is magnesium block is there. Okay, glutamate is coming here again. So once magnesium is going away, from here what will happen? Calcium will come. Okay, now this calcium will do what? It will stimulate colmodulin kinase. It will stimulate what? Colmodulin kinase. Okay, so colmodulin kinase, whenever this colmodulin kinase is stimulation is occurring, okay, so what will occur? Means stimulation means what I mean to say? Phosphorylation. So as a result of this phosphorylation, ultimately what will happen? Protein will be formed. What is formed? Protein is formed. Okay, now this protein will lead to formation of more and more these receptors. All these receptors level will increase. Okay, not only that, from here it is also causing stimulation of presynaptic terminal, especially neurotransmitter like NO is helping in this process. Okay, so along with that, ultimately long-term potentiation means what? Ultimately you are getting a more event, more response at the level of where postsynaptic level. So these are the mechanism with the help of NMDA and AMPA receptor. Okay, so this is occurring when there is a high frequency stimulus. Okay, so here I will just explain it properly. Now I will not be labeling it. Presynaptic terminal, postsynaptic terminal, AMPA receptor, NMDA, NMDA receptor, glutamate will come, then it will bind here, sodium will come. What will happen? Depolarization will occur. It relieves magnesium block. So magnesium is going away. Okay, so beside that, then glutamate come here, again calcium will come, then it will stimulate that enzyme. Okay, as a result of that, ultimately ultimately more and more response. Okay, ultimately more and more response will occur. Okay, so that is known as long term potentiation. Okay, so here beside that, okay, what is playing the role? Some neurotransmitter release. Because of that, it further stimulate presynaptic terminal. Okay, so not only that, this calcium colmilidin, it is also helping in formation of new protein, or you can say it is also helping in long term changes like structural changes. Okay, so this is mechanism in hippocampus, that is Schaffer collateral. Okay, so this mechanism I will explain you again now. Okay, so this is the see here, glutamate was released. So this is what NMDA receptor. Okay, so NMDA receptor, and this is what AMPA receptor. From AMPA, sodium has come. Okay, so as a result of that sodium, what has occurred? Depolarization was occurred. Okay, so here magnesium was there. See, magnesium has been removed. Okay, so calcium is coming. Now this calcium, okay, calcium will cause various effect. Like I have told you, this, it has stimulated this, and it is again causing more and more transmitter release. Okay, other neurotransmitter nitric NO is also playing role in this. Okay, then see how it is stimulated. If low frequency stimulation is there and high frequency stimulation is there, response will be different. If low frequency is there, response is different. Protein phosphatase is released, and as a result of that, what will happen? Long term depression is occurring. Okay, so this is the mechanism for long term potentiation and long term depression.